Thanks, you guys, for coming back to the Soil Matters. Dr. Gerald Pollack, thank you so much for coming, my friend. You are an
It's my
amazing
pleasure.
man with amazing knowledge. I'm going to jump off and let the host take over. So have a great show.
Well, sir, it is a true honor. I've been a big fan of your work for years, although I have lost track of where you're at this point. Kind of gone off some deep ends myself into soil biology, chemistry, and just trying to keep up with the interactions it's been a challenge to say the least. But still, it's an absolute honor to have you here today. And I'd love to have you just first explain how you got on to this or how you started the work, and then kind of bring us up to speed as to where you are now.
Okay, how did I start? Well, I've not been studying water for my entire career. I was actually studying muscle contraction at the molecular level, how the muscle proteins, mostly actin and myosin, how they interact with one another to produce force. There was a theory. This theory still exists, put forth by a distinguished and famous Nobel laureate whose name was Sir Andrew Huxley. And Huxley was a famous guy. He was a member of the Huxley family. He won a Nobel Prize for earlier work. He was knighted by the Queen, Sir Andrew Huxley. He was president of the Royal Society. He was master of Trinity College, Cambridge. And when he walked into the room, there was a hush. It was as though God walked in, and so everybody pay attention. And we had evidence that his theory was bankrupt. It didn't make any sense at all. Didn't fit the evidence. We had a lot of evidence that it didn't work, and mostly I came to realize that not only his theory but other viewpoints made the presumption that all of those proteins inside muscle were operating in a vacuum. Water was nowhere to be found. You'd never find any mention of water in any theory of muscle contraction, and that seemed absurd because because a muscle is mostly water. It's roughly like other organs in the body, roughly 2/3 water. And if you translate that into the number or the fraction of molecules, in other words, if you take the muscle and line up all the proteins or line up all the molecules and start counting, more than 99 out of 100 are going to be water molecules, because the water molecule is so small. And if you think of how many you need to make up that 2/3 volume, it's a lot of water molecules. So it seemed preposterous that the most prevalent molecule sitting inside the muscle, there was a pretense as though it doesn't exist. Doesn't make sense, right? I mean, so at the same time that I began thinking in that realm, I got an invitation to go to a conference, and the conference was to commemorate the life of a guy named Ernst. Ernst was a well-recognized biophysicist, a Hungarian guy, and he'd passed a couple of years before that, and the idea was to have a symposium in his honor. So there were people who represented each one of his two areas of expertise. One was muscle, one was water. So I was the one who was invited to present on water, and of course I presented on water, and I think the reception was okay. But after me came presentations on water, and the main presenter was a guy named Gilbert Ling. I'd heard of Gilbert Ling before, but not very much. Anyway, Gilbert presented his ideas, and his idea was that inside the cell, be it animal cells or plant cells or whatever cells you like. The water was not liquid water. It's not the stuff that I have here. It actually, he said that, and presented evidence, of course, that the water molecules were lined up like soldiers at attention. Now, in liquid water, that's not the case. At least as people understand liquid water, the molecules are randomly oriented and they're bouncing around a fierce number of times each second, or millisecond, or even femtosecond. So this is directly at odds with the current view of how cells work, because the current view that you read in any textbook is that it's liquid water that's inside your cells. And if it's liquid water, you begin to build on the concept that all of the solids inside the cell, which are doing the work of the cell, and at least in theory, they can move around freely. But in this kind of water, they can't. This is sort of like a crystal that exists inside. And it was not only Gilbert Ling who made his presentation, but perhaps a dozen or so people who had evidence to support the point of view of Gilbert Ling. I was really impressed. I thought, my goodness, if this is right, everything else is wrong. Basically, all of biology, everything we learn, everything in the cell biology book or biochemistry book, it can't be if this is right. So naturally, it you got my attention, and lots of things capture my attention. I don't always have confidence that I'm intrigued by what should intrigue me, and so I took one of his books. By that time, I think he published 3 or 4 books. By that time, I think 6 or 7 before he passed at age 100 a year or 2 ago. And I gave it to some of my students to look at, some of the students who I know might be involved. And the feedback was the same from all of them. This is so important, they said, and it looks like he's got quite a bit of evidence that, he might actually be right. And so gaining support from my students and such, I felt I was on a good course, and I scratched my head. That's why I'm missing a lot of hair. I said to myself, what can I do? What can I do? To explore this. Ling was not a popular guy. He was on the fringe, and people on the fringe don't really get a whole lot of acceptance. They get derision mostly, and that was the case for Gilbert. So what I decided to do first is write a book. Why would I write a book? Gilbert Ling already wrote 3 or 4 books on this stuff, presenting massive amounts of evidence. And the reason I thought I'd write a book is that Gilbert's writing is not easily penetrable. You might say it's virtually impenetrable. If you have expertise in all the areas he's talking about, you can read through quite readily, I suppose. I don't have expertise in all those areas. But if you don't, it's a real challenge. And I found out that Gilbert Ling, the genius that he was, he'd sit down at the typewriter or the word processor, and he'd bat something out, and he'd send it off to the publisher. The word editing seemed to be like a foreign language to him. He never heard the word editing. Why would he want to edit? It just and so I decided that what I'm going to do is to write a book that describes the ideas of Gilbert Ling in a way that perhaps is understandable, because it's so important. People need to know about it. They shouldn't be struggling their way through. They should be able to read. If the ideas make sense, it should be easy to expose in a way that's understandable. So that was 2001. My book was called Cells, Gels, and the Engines of Life. And Gilbert Ling hated it. He hated it because he felt it sort of stole his thunder. and that certainly was not my intent. But Gilbert wanted his name mentioned in every paragraph, if not every sentence, and the book was essentially his work, and it was presented exactly as such. So we suddenly Gilbert became my enemy, and it took me a decade at great attempts to restore our friendship, which I finally was able to do when we at a conference presented really clear evidence for the long range ordering of water. He was practically jumping out of his seat with excitement, and suddenly we became friends again. Anyway, that's how I got onto it. Of course, as soon as I published the book, we wanted to do experiments, and we started to do experiments, and we found. I mean, I'll tell you about it, but I just should mention that essentially, we found that the idea of structured or ordered water, we could affirm that that was correct, but we found some differences that are substantial enough that they're really important, and that's when I wrote the second book. It's called The Fourth Phase of Water, and that book has become enormously popular. If you look at reviews, for example, on Amazon, a lot of people are really interested in this stuff, and it's gratifying. It took a while to write it. In my vocabulary is the word editing, and editing is necessary if you want something to achieve clarity. So anyway, I gave you a long answer to a short question. That's how we got involved. So we've been doing it. We switch fields from the molecular mechanism of muscle contraction to the structure of water and what it all means. And it's been well, nothing short of exciting is not the right word. I can't exactly say what is the right word, but I tell you, I wake up in the morning so eager to go on and do whatever the day demands or requires. So that's how we got into it. I think I should stop there because I took up too much time to give you the ancient history.
No, that's exactly what we're looking for. It's a foundational, a starting point, and and I think it's important that people understand how you ended up there in that fascinating new realm of understanding water. And just for the sake of the audience, can you kind of briefly explain, or take your time, we got plenty of it, about structured water? Because this has always been a point of contention among a lot of people as to was Rudolf Steiner real? Is it important to stir in different directions? So if you could just speak about that for a couple minutes.
Well, I can't tell you if Rudolf Steiner was correct. I will get to, if we have time, to a point of that Rudolf Steiner was mentioning about the heart. He said the heart is not a pump. Well, I'm not sure I agree totally with that point of view, but we have a lot to say about the heart, and if we get to it, I'd be excited and happy to talk about it. But
we'll definitely get to the heart.
Well, sure. Okay, if you like, it's part of the heart of the story, so to speak. But okay, so what did we find, and what's interesting, and what? Well, I guess I can encapsulate, before I go into a bit of detail, encapsulate it in saying that water actually contains energy. We don't think of water containing energy, but we have evidence that water contains energy, even direct evidence for that. And how does this come about? Well, first thing, one of the findings that I think is really important. We started doing experiments based on Gilbert Ling's ideas. We didn't know any better, and we were novices coming from a different field. And Gilbert Ling's idea that the water is inside the cell is structured means it's like a crystal. Now, crystals have a tendency to be pure. So, for example, ice. So if you take water that has contaminants in it, the water will push out to become ice. Ice is a pure crystal. To gain purity, it's got to get rid of all of those impurities. Otherwise, it wouldn't be a pure crystal. So as ice forms from the water, it pushes out the contaminants, solutes of various sorts. And so we're thinking, we're looking for somehow a preparation that contains water where particles or molecules are pushed out. In other words, they're not there. And we found it quickly. We found it right next to, we started using gels. And we put the gel, it's a chunk of gel, into water, and the water contained particles, microspheres, little tiny spheres that are suspended in the water. And we looked in the microscope, and we saw that there was a region right adjacent to the gel that contained no microspheres. And we watched it, and progressively that region grew bigger and bigger. It kept pushing out microspheres, and it wound up to be a very substantial size. We thought, maybe this is what Gilbert Ling was talking about. Maybe this is a region, a liquid crystalline region where there's order, and because there's order, it gets rid of particles. And we spent several years looking at all different kinds of properties, and indeed, it was true that the properties of this kind of water, we called it first exclusion zone water because it excluded. So exclusion zone, easy to remember in the US anyway. EZ doesn't work as well. EZ. And later, when we knew more about it, we realized that's not a great name, although it stuck. Fourth phase is maybe more appropriate. So one of the principal findings was that this region was not neutral. Now, Gilbert Ling's idea that water molecules are organized, water molecules are neutral. You can stack water molecules, from here to the moon, and you'll never get negative charge. But we found experimentally, by sticking electrodes directly into this region, that it was negatively charged. So Gilbert Ling, unfortunately, couldn't be right in the exact interpretation of how this structure is, and since it had negative charge, we looked for, you're starting with water molecules which are neutral, and you find a region of negative charge, and therefore, it goes without saying that you need to find another region with positive charge, because the positive charges can't just vanish. You have to have an equal number of positive charges as negative charges, because it all starts from neutrality. So we found where the positive charges were. They were immediately adjacent to this exclusion zone. So you have a gel that's right here, and next to the surface of the gel, you've got an exclusion zone, negative, and beyond that, in the water, ordinary water, you've got a lot of positive charges, a lot of protons or hydronium ions, if you will, which means you got a battery. You got a battery, you'd think, well, hey, wait a second, this is a battery made of water? You can demonstrate that it's a battery. You just take two electrodes, which we did, one in the negative, one in the positive, and you connect it to a load. We connected it to a LED, a light emitting diode, and voila, you can light the LED. So demonstrably, it means that it's got energy. The water somehow breaks up into negatively charged exclusion zone fourth phase water and positively charged ordinary water. And okay, so that's different from what Gilbert Ling had found, and we were naturally excited to find that. However, the question arose, if you got a battery, everybody knows that batteries need to get charged, right? You can take your cell phone, and if you forget to plug it in during the night, the next morning you can't communicate with your cell phone. So, but this is a generality. Whenever you have a battery, you need to charge it or recharge it or whatever. So the question arose: well, where does the energy come from? And we couldn't figure it out. That's another reason why I've lost a lot of hair. I was scratching, couldn't figure it out. And one day, it was an undergraduate student who was working in the lab who figured it out for us. So he's sitting at the lab bench or standing at the lab bench. This was a tall bench, and he had a chamber with a gel in it or polymer. I don't remember. I think we began using the polymer Nafion, which gave rise to very nice, stable exclusion zones. So we used it a lot. And he's playing with it, looking at the exclusion zone. And I don't remember what the intent of his experiment was, but he found a lamp right next to him. It was one of those gooseneck lamps, and he picked up the lamp and he shined it on the chamber. And he called me in to see what he'd found. And what he found is that the region of the exclusion zone that was illuminated, it grew like crazy. So I came in and I said, wow, this is really quite an observation. Why don't you remove the lamp or turn it off and let's see what happens? And so he did. And that region that had greatly expanded retracted back to its initial. It took some tens of minutes, but it looked like it was reversible. So it didn't take a rocket scientist. This is middle school stuff. You shine the lamp, light, and light is building it. So maybe light, photons are responsible for the energy. And of course, you think about plants then. You think about photosynthesis and such, and indeed, I think that what we found is perhaps directly related to the first step of photosynthesis, the splitting of water's charge into positive and negative through light. So it has direct relevance, I believe. Anyway, we found that the battery is charged, and in our experience, we of course wanted to find out, well, light has many wavelengths. Which wavelengths are we talking about? So we did real experiments, and we checked it a wide range of wavelengths. At the short end, starting with ultraviolet, through the visible spectrum, on to the longest wavelengths, infrared. UV did nothing. Actually, it does something we found very recently, but it didn't grow the exclusion zone. Visible, same thing, except when we began to get to the long wavelengths, to the reds. The reds, we found the reds tended to build the EZ a little bit, but beyond that, longer wavelengths, the infrared, wow, it was like gangbusters. A very, very small amount of infrared light, infrared energy, hugely expanded the exclusion zone or the fourth phase. So we knew that this is really, really important. And most people don't understand exactly about infrared. Most people think, well, where do you get infrared energy? You kind of think, well, you turn on the range, and the coils on which you put the pot, they glow bright orange and they generate heat. And people say, well, that's generating infrared energy. And it's true. However, infrared energy comes from virtually everything. So if I look at you people on my screen, if we turned off all the lights in your rooms or studios so that you couldn't see anything, and your cell phone camera couldn't record anything, if you whipped out an infrared camera, which is basically like a regular camera, except the sensor is sensitive not to visible light but infrared light, you get a beautiful image. And that's why this is used for night viewing, for example, by the military. because everything is generating infrared energy. And because everything is generating infrared energy, you can see what's going on, but also that infrared energy is available to build easy water. Therefore, almost always, if you have water and the right kind of nucleating surface, we found it's gel surfaces and polymers, biological surfaces, they need to be hydrophilic or water loving, and then you get easy. So you got the energy available, and therefore if you got the right kind of surface, which is most surfaces, not all, it means you're going to have some easy water. And the more infrared energy you have, the more easy water you'll have. But there's always some, if the situation is right. And also inside your body, you're generating heat from metabolism. And that heat also is essentially, it's not exactly the same, but essentially the same as infrared energy. So in terms of you and your bodies, you got lots of water, you've got surfaces that generate easy water, and you've got energy, infrared energy coming from inside of you and outside of you. So you're going to be full of infrared energy, full of easy water, I'm sorry. And of course, the same is true of plants. Come springtime, when it gets warm, when infrared energy becomes really abundant, easy is going to grow. And so the cells that exist inside the plants are also filled with easy water. And I didn't say this, but I treat this in the first of the two books, The Cells, Gels, and the Engines of Life. The way cells appear to function is when cells are in their dormant or relaxed or not doing anything state, the proteins inside are in their elongated state, and the water is in the EZ state. Now, nothing can happen when the water is in the EZ state. It's sort of like embedding something in ice. It's not ice, but it's crystal, just like ice. Now, if the cell wants to do something, for example, if a muscle cell wants to contract, or some cell wants to shorten, so the plant bends or elongates or whatever, the water needs to convert, and it converts. I marshal a lot of evidence that when the cell goes into action, the water converts from EZ water to ordinary liquid water, where things can move around, things can happen. And then when the action is over, it returns back to the initial state. And that, by the way, is the energy requiring state. So that I just wanted to mention the energy, because energy is really important. And well, I think maybe I should stop there and wait for your next question, because I can go on and on and on. But
take a drink too. Moisten that mouth. You've been doing a lot of talking, my friend.
I will take a drink.
So you talked about the negative and positive charges, which is really interesting. So in a cellular, inside of, say, just any cell, usually have an inner and outer wall. Is the easy water pushing the protons out to the outer cell wall so that it gets that division?
Yes. So what happens? I guess I've thought mostly about animal cells or humans rather than plants, but I think the principle is going to be the same. So what happens with us is we start with water, which is neutral. Now the water transitions into EZ water, which typically is negative. And what about what happens to the positive? You might think, well, they're just outside the cell, and basically there is, because the cell is negatively charged. This is true in animal cells as well as plant cells. Plant cells are typically minus 100, minus 150 millivolts. Animal cells not quite so negative. And so your question is, well, what happens to the positive charges? Well, in animals, first well, let me put it another way. Every time you breathe out carbon dioxide and you breathe out water. Water plus CO2 gives you carbonic acid, H2CO3. Carbonic acid, it's got lots of protons, it's an acid, and so those protons get exhaled. So with each breath, each exhalation, you get rid of protons, you get rid of positive charges, and we remain negatively charged. So it's not, and the plants as well as the animals, this is true. So what you have inside the cell is you've got a lot of negative charges, and that I argue, contrary to prevailing views, which deal with channels and pumps, it's the negative charge of the easy water, I have argued, which is responsible for the electrical potential of the cell. And I believe that's true. And, you know, if you want evidence, I can talk about it. So that's the reason our cells and plant cells are negative. Now, where does the energy come from? Well, if you got a cell that's filled with negative charges, you ask the question, well, what do negative charges want to do? They want to get away from each other, right? They repel. And that proclivity for those charges to escape from one another, that's potential energy. You know, you've squeezed all the negative charges together and they want to separate because they repel. So the fact that they're squeezed together inside the cell, that is the energy we're talking about. It's potential energy, and it's important for the plant and important for animals. So returning to the plants, which I know is most interesting for you people. You know, when spring comes, lots of warmth and lots of infrared energy, which means that easy water is going to build, right? And we found something that I believe is directly relevant for plants. You know, a question has always been in the xylem of the plants. Xylems are conduits that bring water from the soil up and up the plant. And some plants, as we know, like redwood trees, are 100 meters tall, 300 feet tall, and this is a mighty column of water. You know, if you took a pipe that's 100 feet tall and you filled it with water, the pressure that you'd feel at the bottom is enormous, right? So, but somehow the water works its way up to the top. Otherwise, the leaves wouldn't be green. And so what's going on there? Well, and there I'll tell you about a discovery made by another student. Undergraduate students are amazing. Sometimes, you know, they're so creative. They'll do anything. Hey, you know, I'll try this or I'll try that, and I encourage them. Always
happy accidents, I think, is how you term that, Gerald, if I remember correctly.
It was a happy accident. Yeah, it was an accident because this particular student, I assigned, I don't usually assign. I discuss a few possible projects, and the student chooses what the student likes to do. It works best that way. So, yeah, happy accident. So I asked him, we have just found that the polymer naphthalene, which we had been using quite a lot, we've been using sheets of naphthalene, and we just found out the naphthalene also comes in tubes, like a straw made of naphthalene. So I asked the student, I said, you know, we know that a flat sheet of naphthalene grows EZs very nicely, but if it's curved, as in this tube, does the curvature impair the ability to grow EZs? So he did the experiment, and he quickly found out, no problem, lots of EZs in there. And I was away on a trip somewhere, I don't know, Europe, whatever, giving lectures, and he he'd already solved the problem. He answered the question, and I wasn't there. And he thought, okay, well, I got to do something. So he started doing experiments on his own. And one day, when I had returned, I was sitting in my office with a visitor, and the visitor was maybe less than compelling. And I was sort of hoping the meeting would end. And fortunately, this same student barges in, and I pretend that I'm irritated because, he barged in and I'm speaking to somebody and he shouldn't interrupt that way. But he said, I got something I thought you'd want to hear. It looks pretty interesting. Okay, so tell me, what is it? I was actually delighted that he interrupted the conversation, but I didn't.
I love it.
I didn't want to demonstrate it. And okay, anyway, he said, I looked at the tube of naphthalene, just like a straw, horizontally situated in the chamber, covered by water. And he said the water kept flowing through the tube from one end to the other, and it wouldn't stop. And he said, I thought it was pretty interesting. I wanted to tell you that. And so I thought, if this is real, and we quickly confirmed that it was real, is really important because it would seem usually what you need is a pressure difference, a pressure gradient from one end of the tube to the other. But this tube was just lying in the chamber like this, a water equal height above both ends of the chamber, and it just kept going. And you need energy to do that. So where does the energy come from? We had just found that about the infrared energy. And I know infrared energy is coming to the chamber. It gets absorbed because water loves to absorb infrared wavelengths. And that energy was getting transduced by the water into something that produces flow. And we began to understand exactly what the mechanism is, but it had something definitely to do with easy water and the separation of charges. So I was, this was really exciting. And the reason I mentioned this is there are two reasons. First of all, it has something to do with the heart, which if you ask me, I'll tell you about. But it has to do with plants too, because one of the questions in plants is what causes flow? There are all kinds of flow in plants, including the one I mentioned before about the xylem and the flow from the roots all the way up to the top of the trees. And by the way, it's especially prominent in the spring and summer when you got lots of infrared energy. And I'm quite certain that the mechanism that I'm talking about, that this student identified in the laboratory, well, he identified the phenomenon, maybe not the explanation, that this is highly relevant and that this is indeed the explanation for all kinds of flows in plants. Not just the xylem, but the phloem and anything else. It's hard to find the source or the reason for the flow, and I think we've identified it. Just to complete the story, if anybody is interested, what happens is an annular region of EZ forms just inside the vessel, outside too, but just inside the vessel, negatively charged. It discharges protons, as I was discussing earlier, as it forms. And so the protons are sitting around the axis, the long axis, central axis of the tube. And those protons are free. They want to get away from one another. And the more protons that are released to around the center line of the vessel, the more pressure you have to get out of there. They want to get away from one another, so they'll exit either one end or the other end. And once they exit, they're pulling the water with them because the protons are actually attached to water, giving you hydronium ions. And so those positive hydronium ions are going out one end and dragging more water in, and it just keeps perpetuating itself. And if you've got energy, if you've got infrared energy, the more you have, the faster it'll go. So So that's with regard to plants, which I know is a subject of great interest here. But also, if I may, I want to talk about the heart. Do we have? Okay, I see a thumbs.
I was segueing you into this.
Okay.
Onward, upward.
Well, because we're really excited about that. We found something that I believe is fundamental. Let me just start with: Did you know that? There are half dozen papers that have appeared over the past 100 years that nobody knows about because it seems oddball, but all the findings are similar, and each one is done in a different experimental setup, different animal, whatever. The heart stops, and you expect the flow will stop too, but the flow doesn't stop. The flow continues, and if the heart's not pumping but the flow continues, you got to scratch your head and say, what the hell is going on here? Because something's not right, and people have not paid attention to these papers. So something's going on. There must be some other source besides the heart that drives the flow. At least, that's the logical conclusion. But it didn't start that way. It started with me with a trip to Moscow. I was visiting my friend Vladimir Vyakov, a guy who's vice chair of biochemistry at Moscow University, which is generally regarded as their -- university. And he introduced me quickly to the guy down the hall, and Vladimir wanted me to hear what he had to say. And so the guy starts off by saying, with translations from Vladimir, saying, there's a big problem in the cardiovascular system. Big problem? I never heard of a problem. What's the problem in the cardiovascular system? In fact, I did my Ph.D. work studying the cardiovascular system, studying the pressures and flows. And I guess I thought I had it all worked out. We could understand the pressure waveform and the flow waveforms and the different regions. And, so I came in with, as the Israelis like to say, cutting the clouds with my nose, a bit arrogant, thinking, what on earth is this guy going to tell me? He says there's a big problem. Well, after 5 minutes, he had me convinced that there really was a big problem. So what's the big problem? The big problem is a plumbing problem. If you think about it, it's nothing complicated. Red blood cells are 6 or 7 micrometers in diameter. They have to pass through blood vessels, capillaries that are down as small as 3 or 4 microns in diameter. So imagine, you've got a big blob that needs to pass through a thin tube. It's like, like your toilet getting stopped up. You got to take the plunger and you plunge and plunge and plunge, and finally you get it out of the way and the toilet's working again. Well, the same principle applies. You need energy. You need energy to take those red blood cells and bend them, squeeze them so that they can work their way through. And indeed, they're squeezed. You can see it on videos as they pass through those narrow capillaries. They're squeezed through, and it's just they've got to work their way through. So these guys, Russians, are really into calculations. They like to do that. They're highly quantitative. So these guys calculated how much energy do you need, and it turned out to be nothing short of astronomical. They computed that if the heart were responsible for providing the energy to squeeze every one of those red blood cells, the amount of pressure that the heart would need to develop would be something like 1 million times the pressure that it actually develops. So you might say that's high blood pressure, but high to the extreme. So it's obviously the heart can't be responsible. There must be something else that works together with the heart. I hesitate to say instead of the heart, because heart, it develops pressure and it seems to drive the blood, despite what Rudolf Steiner was saying about the heart not being a pump. I don't know, it depends on your definition of a pump exactly, but it's got to play some role. But in addition to that, there's got to be something else. So what could that something else be? Well, hit my head over my head with a hammer. We just found in the laboratory that you take a tube made of naphthalene and put it in the water, a hydrophilic tube, and the water, quote, spontaneously flows through it. We know it's not really spontaneous. It has to do with easy buildup, which is then fueled by infrared energy. But I thought, maybe the same thing applies in everybody's cardiovascular system. Maybe the vessels themselves are driving the flow. And so we went ahead and tested it with positive results. And the guy who did the hands-on test is a student of mine who now got his Ph.D. and moved on. And he told me afterward, when I first proposed the idea to him, he said he thought I must have been on some kind of drug because, preposterous. Everybody knows that the heart is driven, that the heart drives the circulation. I thought so too. Why would anybody have reason to doubt it? But as I've told you, there is reason to doubt, especially the idea that, if you stop the heart, the flow continues at a low value, but it continues. So something else must be pumping. So he did the experiment. He did it on a chick embryo. So living circulation, a 3-day-old chick embryo, the circulation is very well developed. But the regulatory systems, the hormonal and neuronal regulatory systems, are not yet well developed. So it's a pretty pure situation. And he developed a way of imaging various blood vessels so he can track the flow. First thing he did was he stopped the heart. And indeed, the flow continued. So there's got to be something else, right? Unless there's some kind of artifact, because 2 + 2 = 4. Very simple logical deduction. Something else is at play. And then he tested the signature feature of what we found in the laboratory, and that is, if you add infrared energy, the flow goes faster. And that's exactly what we found. And you take away the infrared energy, it returns back to where it was. So we got that result, and the paper has been submitted for publication. But we think that the results are sound, and the bottom line from this is we all like to think that our circulation is driven by the heart, but we found that it's driven not only by the heart, but also by the vessels themselves. For technical reasons, we can't say what percentage is driven by the heart and what percentage by the vessels, but we have reason to think that it's pretty substantial. The vessel-driven component is quite substantial, and it'll be in the future that we attempt to find out what the percentage actually is, but it's probably substantial. So this is kind of fun and neat, out of this simple incursion into studying water and energetics and whatever, we come forth with a finding that has pretty important implications. So that's one of the implications, and let me stop there because I think you guys have more questions.
Do you want to take a question, or because I got another one that I think is really all right. So, did you ever try increasing the diameter of the naphthalene tube to see if the flow increased?
Well, we did try increasing the diameter of the naphthalene tube. By the way, we used other tubes. We created we created tubes. We took a gel, and as it was gelling, we put a wire in, and you pull the wire out, and you're left with a chunk of gel with a tunnel, which is sort of like a tube. And you immerse it into suspension of water with microspheres, and essentially you've got the same story. And we found that we used more than a half dozen different gels, and we found the same results. Sometimes it's faster, sometimes it's slower, but basically the same result. So it's a general result. Yeah, we did. But if you think about it, when you increase the size, of course, we were eager to see what happens. You don't get what you might reflexively think. You think that the larger the diameter, the more hefty the flow. It doesn't turn out that way, and the reason is the size of the exclusion zone. It doesn't. It increases, or I should say, it's got a maximum size typically. Depending on the material that you use, the nucleated growth, it's typically 200 micrometers or 300 or 400 or 500 micrometers on that order. That is like maximum half millimeter. We've seen larger, but essentially. So you can make the tube a diameter half a mile, and you'll still get this 500 or 400 micrometer size annular exclusion zone just inside the tube. And then, as I said earlier, that as the EZ forms, protons are generated, and those protons go into the core of the tube. And they're not going to be very concentrated if you have a large tube. And therefore, you want them to be concentrated, because the more concentrated they are, the more they want to get away from one another, and the more brisk the flow. So the whole thing almost fails when you exceed a certain diameter. There's just not enough proton pressure to drive the flow. There are ways of getting around it, and we've actually explored ways. You can basically put a whole lot of tubes in parallel and increase the flow that way, or simulate the tubing in different ways, and have multiple channels and increase the flow. I think that's what you were getting at. So, long answer to a short question. There's, at least in our hands, there's a maximum diameter, beyond which, if you go, you don't achieve anything.
All right, Ab, do you want to fire one, or should I throw another one at him?
Oh, let the man have a drink, okay?
Okay.
Gerald, have a drink of that good water that you got beside you.
Thank you.
I'm going to jump in. I mean, this is phenomenal, and Jerry, your work had helped me pull in so much stuff. Probably some of it wrong, but some of it just, bringing it in, whether it's better understanding of soil microbiology or even better understanding how nutrients come into plants. But I'm just wondering, what is because I know you've made some reference that easy water could potentially have memory.
Yep.
But how quickly, how stable is easy water? Is it something that's changing like that, or can it have stability?
The answer is yes. That's sufficiently vague. Well, we haven't explored that question in great detail. However, I can tell you of at least one anecdotal kind of observation. Some guy called me. This was a decade ago. And he called me, and he told me he's been producing a kind of water. I don't like to mention names or products or whatever, but he said that it has a tremendous curative power, and and he told me actually it can cure. He had some neighbor who knew somebody who had irreversible kidney pathology. and she started drinking the water that he was producing, and 1 month later she had no kidney pathology. So I said to him, I don't believe you. I actually did believe him, but so he said, oh, I'll send you the hospital records. So he sent me the hospital records, and it confirmed exactly what he said. And so I wanted to, at that time, we were a little bit more open and willing to test other people's waters. Now because if we did, so many people want us to test their waters, and we just don't have the people power. And if we start testing waters, that's all we'd be doing. So I have a generic answer, which is sorry, we can't do that. But anyway, I wanted to test his water to see if it contained EZ, because I thought at the time, and think even more so now, that if you drink EZ water, if your cells are absolutely filled with EZ water, and the same with plants, if they're filled with EZ water, they're going to be healthy. And so we were busy, and a sample of water that he sent sat on my desk for 3 months. We tested it after 3 months, and we have a simple test for EZ water, and it contained a substantial amount of EZ water. So what does it prove? It doesn't prove too much, because it might have started with a huge amount of EZ water, and it declined over a few months to the level we measured. Or it could be that had no EZ water and just sitting on my desk, somehow it acquired some. Or it could be stable over a period of, so we don't know the answer. And it's not so easy to do experiments to check that, but it is possible, and we intend to do it. Of course, one of the issues, as is true with so many, especially people who work in areas that are not how do you say, settled science or conventional science. It's really hard to get money, and we need to carry out experiments like that. We need people, and to get the people, you have to pay them, postdoc or graduate student or something, and it's really hard. And the more our work deviates from mainstream norms, the more difficult it gets. We had a funder, a wealthy individual, who found us and said, "Hey, I like your stuff. I want to fund you." And he did so for I think 6 or 7 years, and he ran into some financial difficulty, and he had to withdraw. So we're now in the position of trying to find support. And it's not so easy to find support. If you're doing something mainstream, it's easy, or if you're doing reasonably decent work. But if you're challenging the mainstream, often if you try to get money from various agencies, federal agencies, and you put a proposal in, typically they're going to get an expert in your field, and those are the very people under challenge, and so it's obvious. It's like if the revolutionaries, French revolutionaries, came to Louis XVI and said, we got a bone to pick with you. You can expect the response that they get. It's essentially the same. So to carry out tests of the kind that you'd like, and there are others of similar value, you need people power. And we simply don't have the money to get the people power. We hope that some people who maybe recognize the value of what we're trying to do will help out, but it's a real challenge, I can tell you.
I mean, my guess is that it is an incredibly dynamic thing, and that it despite forming crystal structures, I think that is changing relatively fast in terms of the way nature would probably use it as a bit of a storage for energy, but then be able to use that energy and replace it every time.
And therefore, sorry, I wasn't following. Therefore, you think it's stable or not?
No, thinking that it's dynamic.
Dynamic. Okay. Well,
there's energies all, and it would help me understand that how water can be a conductor, right? Because if it didn't, it wouldn't conduct as well if there was a zone that did not have that charge. Right?
Well, yeah. So the question is, what is the conductivity of this material of EZ? And it's not so easy to measure or to decide what it ought to be. If you've got electrons in there and the zone is typically negatively charged and those electrons are freely movable, it might be that it has an extremely high conductivity. But if everything is stuck and immobile, then it would have low conductivity. So I think these experiments actually do need to get done. It's just like the experiments you're talking about the stability. The stability depends to some extent on which state is more stable. Is the EZ state more stable, or is the liquid water state more stable? And the answer is not so clear. Probably there's an energy barrier between the two of those. And once you cross the energy barrier, you're down at a lower energy level. But which one is higher, which one is lower? You can debate that, but the answer is not so clear. And that will have a lot to do with the longevity of the EZ water.
And Leighton, do you mind if I ask one more question? I love the work. I believe perhaps a graduate student, but somebody from your lab, Abha Sharma, did some interesting work around turmeric, I think coconut water, Tylenol, and Roundup.
Yep.
Do you mind sharing a little bit of that
Sure.
That work with?
Sure.
And I also wondered, did you do any work with cannabis?
Yes, we did, but we were stopped because we didn't have a license to do it. It was stopped right in the middle, and we finally got a license. It took 2 years. The student who had been working on it quit or graduated or whatever, and we haven't gotten back to it. Simply, we need people power, and it's on our agenda to do.
Yeah.
So, we tested. There are various agents, very well from Ayurvedic times, and from ancient China too, that have been known to be health promoting, used for many years. And the star of the show is turmeric, but there's also holy basil and coconut water and various other agents. And we wondered what's the mechanism? How come they promote health? And of course, being into easy water and such, we thought one possibility is that those agents build easy water. And if they build easy water, it means that we have more easy water in our body. Our cells are filled with easy, and so we tested it, and we had one student test each, one student to one compound, and they all found pretty much the same thing. And Abha Sharma was the sort of general director, experienced scientist. We found that it built easy water. These substances built easy water, and it built it in a fairly wide concentration range, the concentration that we might expect to have in our body. So we could draw the conclusion that these agents, known to promote health, that the reason, or a possible reason, that they promote health is they build easy water, because the more easy water you have in your cells, the healthier you are. I didn't go into the evidence for that, but if you're short of easy water, your cells are not functioning very well, because in order for your cells to function, the essence of function, as I mentioned a little earlier, is you start with easy water, and the easy has to undergo a transition to ordinary liquid water and then back again. But if you don't have enough easy water, can't do it. So the cell needs to maintain a substantial amount of easy water. And that's exactly what turmeric does. It enhances, builds up easy water. The same thing that basil does. And probably many more agents that we haven't had time to discuss. But as a control, we decided to check a poison. Uh, and well, Roundup is is the classic poison, and so we tested it, and glyphosate being the main ingredient. And we did it exactly the same way as we tested turmeric or basil. We added it in increasing amounts, and we found that at no concentration, starting from zero all the way up, at no concentration did it enhance or build up easy water. It always diminished the amount of easy water, which means that it essentially dehydrates the cell, because easy water is essentially hydration. So that was a control on our part. We expected to get a result similar, and that we got that result. And we don't know. We'd like to test some more poisons. I always hesitate because a lot of these experiments are done by young undergraduates without a lot of experience, and I don't like to expose them to dangers like that in doing the experiments. But the experiments do need to be done. It could be, this is a total speculation, it could be that many poisons are poisons because they remove easy water. They diminish easy water. We don't know that.
And they all have to be hydrophilic, right?
When you say they all, what's
like everything that you were testing, does that have to be hydrophilic, or can it be?
Well, it should be soluble in water. Yeah. Or there are some tricks that you can do. I mean, essentially anything that you can ingest. I'm not. So I don't know how familiar you are with all the agents that date back 5,000 years from the Ayurvedic tradition, but they were smart guys and smart people there. They knew what they were doing, and everybody, even 5,000 years ago, wanted to be healthy, and so these developed over time, and it's a total shame that so many of us are ignoring that, because it works, and we think we understand now how it works by building easy water in the cells.
Would you classify easy water as an antioxidant? Does it function in a similar way?
Well, it's got negative charge, and I guess I hesitate. We thought about that, like a dozen years ago, but I don't recollect all the evidence. So may I pass on that one?
Yeah, sure.
Yeah. Okay.
I'll turn it back to Leighton now. Thank you.
Okay. Yeah.
So what you're saying about poison really explains a lot of what we see in soil biology. That the cells, or the organisms, the bacteria that can actually survive in those very toxic environments tend to be very limited in diversity. And so what makes sense is that it literally is killing off the most advanced as well as a lot of the diversity of the biology. And it only makes sense that it's doing the same to us when we ingest it. So I think that's a huge indicator of the fact that it's more toxic than we're even talking about, as far as, whether it's an herbicide or a pesticide or any of these sides, right?
Well, yeah, totally. Absolutely. Toxicity is well recognized. The champion of all that is Stephanie Seneff. I'm not sure who has studied glyphosate more deeply than anybody on the probably on the face of the earth, and understands the principles very well about
Okay,
all
I'm
this.
going to jump in. Can you say that name clearly so that we make sure people get that name so they can do the research on her as well, please?
Stephanie is the first name. Seneff, Seneff. I'm not sure if it's one N or two N's, from MIT.
I'm sure Ken will find it. But,
Yeah, Stephanie Seneff. She's collected evidence. She operates intellectually at roughly 3 times normal speed, and she knows every paper that's been produced, and the evidence is massive about the destructiveness of all that. So yeah, I highly recommend anybody who's interested in glyphosate to just Google under her name, and you'll find huge volumes of evidence. Yeah.
all right, so this is a deep one.
Uh-oh.
Yeah, you think that it can go the other way too?
Well, I'm sorry. What do you mean?
Energetically, instead of being electron, more proton. In other words, here's my reason for asking that question. There are a lot of bacterium, archaea too, that can live in very, very hostile environments.
Yeah.
With these poisons. Like, I've actually harvested grasses that have been exposed to 60 plus years of being exposed to these horrific herbicides. And I extracted the soil and the soil bio, and I separated out the soil biology, and then applied it to an area that had been devastated by Roundup. And what I noticed was that within a matter of weeks, I treated the soil, gently watered it for a couple weeks. Then I overseeded it, put some hay on it, and then put a biological inoculant on it, and it took off. So why I'm asking that is I wonder if, this Roundup or glyphosate, whatever you want to call it, that is killing a lot of the diversity that I see. Is there another one that actually can convert it and use it as an energy source? Because that's what it appears that it did. So wouldn't that kind of lead to, well, if glyphosate kills the easy water in certain bacteria, but it doesn't in others, that bacteria may actually be the opposite of easy water?
I can't answer that because I don't know enough, but generally in science, the principle of simplicity, and simplicity would, if the principle applies, is called Occam's razor, after Sir William of Ockham, I think the 12th century or something like that. Simplicity is the essence of science, and so you'd expect that if glyphosate worked in one way in one plant, you wouldn't expect it to work the opposite way in another plant. I guess I could speculate that the competition is killed off. Maybe some other species are more sensitive to glyphosate than the ones you're talking about. So with the competition for nutrients killed off, maybe the species in question can flourish. I mean, that would be one possible interpretation. I don't know enough details to comment beyond that, but I would be surprised if glyphosate is so broadly speaking a poison. I wouldn't expect it to do the opposite in one particular species. Well, it's just my speculation. I really don't know anything about the particular circumstance or the subject, so I shouldn't speculate beyond what I know, a small amount.
Well, I'll be honest. A lot of the work I've done was speculative, and then came up with interesting results. And the only reason I wanted to ask that, and I hope I asked it correctly, because I was talking about the bacterium that were, because I did an extraction of the soil biology to treat the soil that had been damaged. So that's what I was thinking. Like, and generally speaking, there has to be a yin and yang in life as well. So theoretically, there would possibly be another opposite, proton charged organism. But again, this is way out in left field. So
well, I can't. I'm not sure. I mean, you could be absolutely right, but I really don't have a lot to add. There's one that's sort of related, but not. We owe Benjamin Franklin um, the honor of having labeled the electron negative and the proton positive. And at least for humans, and I think for plants too, negative charge is really important. The more negative charge we have, the healthier we are, and the more positive charge we're subjected to, the less healthy we are. And there are a lot of evidence all over the scientific map to support that. So it turns out, that negativity has a positive effect. If Benjamin Franklin had reversed the nomenclature and said that let's call electrons positive and protons negative, then we could say a positive charge has a positive effect. It would be a lot easier and more sensible instead of the opposite. But I guess we, if we pay homage to Benjamin Franklin for his important discoveries and such, we can't really challenge him on that.
no, it's a little late for that, I would say.
Yeah, that is true.
Another really interesting thing about the heart. So we all know you can't compress water, and the cell is what percent water. So when the energy, the positive charges around the artery or the capillary are helping to push that platelet through, is it twisting because you can't compress it, or is it just elongating to get it through that tighter space?
Well, the pictures that you see, I mean, this is direct evidence. You can see it on videos. The red blood cell, which is really what we're talking about mostly. Red blood cell has the shape of a kind of donut. Although it's not a complete hole in the middle, it's a donut with some filling in the middle, and it looks as though you're taking this planar donut and bending it this way. So you can imagine, if you have a flexible donut and try to push it through a vessel of some sort, that's the way you'd expect it to bend. And it's a fair amount of energy that is required to do that. And also the other issue is that there must be some reason for it. Nature, yeah, there you go. So they're red blood cells, and they actually will bend around. If you take a midline and draw a line between the two halves of the cell, they'll bend around that line. It's what the videos show. I don't know how much energy is required, but you can imagine it's a fairly substantial amount. And doing calculation, of course, all calculations require assumptions, so you can never be sure. But I was totally shocked when these guys were telling me it's about 1 million times the pressure that if the ventricle were responsible, it'd have to generate 1 million times the pressure that it actually generates. It's a lot of energy. So if you think along those lines, if their calculation is even near being correct, even if it's off by a couple of orders of magnitude, it's still a huge amount of force, of energy that's required, which implies that the ventricle may be responsible for only a very modest amount of the energy that's needed. The rest coming from the cells themselves. From the vessels themselves. So it could be very substantial. I'm sorry, I deviated from your question.
No. That was my whole point, was that the amount of energy that's in the body is clearly, as you said, the heart would have to beat 1,000,000 times harder to or create more pressure.
Yeah,
That's got to be pretty damn significant.
that's very significant. Yeah,
right. And so I recently saw a paper talking about millivolts of a cell, and they said, if you add up all the trillions of cells in our body, and you put them together, then in many ways the human, and I'm sure animals too, are in many ways generators of electricity.
Exactly. Yeah. So, we're electrical generators. We run on electricity. The biochemistry books and cell biology books presents us as chemical beings. More and more, it's pretty clear that we're electrical beings. So let me step back. So was it 70 or 80 years ago? The idea of ATP was discovered, and the idea was that adenosine triphosphate, one of the phosphate bonds of the three of them, is a high energy bond. And that high energy bond, is used to confer energy to whatever the body does. That's the ultimate source of energy. Well, a year later or 2 years later, someone challenged that point of view. And I hadn't known about it, but Gilbert Ling on his website, gilbertling.org, it still exists, tells the story of that. So another group said, the first group who said there's a high energy bond, they made a simple arithmetic error. They screwed up. They're wrong. It's not true. Well, since then, so you have a hypothesis, and then you've got another group that says you're all wet. You're wrong. There is no such thing. And nobody has ever followed up on that. So in truth, we really don't know who's right. Is it the original perpetrators of this idea, or is it the challengers who said there's nothing to it? We don't know. And that demands further exploration, because
that is absolutely mind-blowing.
It's mind-blowing. Yeah, it might be all wrong, completely wrong. I don't know if it's wrong or right, because the experiment, or it needs to be addressed by somebody. And so what I'm putting forth, if it turns out that it's wrong, and I don't know if it's right or wrong, there is another form of energy. It's the one I've been talking about, the electrical energy that comes from the water. You see, and there may be a lot of energy that comes that way, because it doesn't come exactly from the water. It comes through the water by virtue of the energy that we're receiving all the time from the environment. And of course, the champion of all that are plants, which get that energy perpetually, but we get it too. And so it might be, again, I use the term might because we don't know. It could be that our energy comes principally or even entirely from electrical energy. In other words, it could be that most, or even all is a possibility of our energy is electrical, which is pretty interesting.
Yeah, it is. It's amazing that no one's really chased that rabbit, because that could change a lot of things.
It could, absolutely. The biochemistry that's associated right now with the production of ATP is very complicated. If you read the biochemistry book and go through all that, you really have to scratch your head and try to understand all the steps that are involved. And well, one likes to think that nature is simple. Nature works simply, but who knows?
I don't think it's as simple as anybody wants to make it out to be.
Indeed, it may not be. Well, we're certainly intricate because we have so many different components inside our body. There's no doubt it's intricate, but the question is about the fundamental principles of how we operate. And I guess I find myself convinced that those principles, essential principles, are going to turn out to be simple, not complicated.
Hey,
It's
Jerry,
so complicated it becomes simple.
I'm sorry, once more.
I said it's so complicated.
Oh.
I'll bring my mic over. It's so complicated it becomes simple.
Oh, well, that's one way of looking at it. Yeah, you could say that. The principle is, is really far-reaching. This guy, Sir William of Ockham, he was talking about the existence of God. He was a theologian, philosophical theologian, and he said, "Hey, there are two ideas here, competing ideas. God exists. God doesn't exist." He said, "The way to find out the truth is put up a hypothesis that God exists and another hypothesis God doesn't exist. And whichever is the simpler one is probably the one that's going to be right." And then his ideas remained dormant for a few centuries. And then Isaac Newton said, "Hey, this guy knew what he was talking about. Let's apply the same to science." He said, we have two competing hypotheses to explain a certain phenomenon. And the simpler one is the one that's probably going to be right. And that principle was taken up by scientists for hundreds of years. And it was actually used, I think, usefully for many centuries until about 100 years ago with the advent of quantum mechanics and such, where we're now kind of led to believe that in order to understand nature, we need abstract mathematics. It may be right, but some question is raised about whether Mother Nature really requires abstract mathematics to produce life or produce nature, and I'm not so sure.
Interesting stuff for sure. One real quick question: the water that was sent to you, was it alkaline?
I don't know that we measured the pH of the water. I can't recall. We may have, but I don't remember the result.
Okay. All right.
I would say probably it was because we simply we need negative charges in our body, and if the water is healthful, it's going to probably have a higher pH. And by the way, there's misinformation. A lot of people think that if you drink water that's alkaline, or so-called alkaline water, which has become somewhat controversial, that it'll get neutralized in the stomach anyway, so why bother? Because of the stomach acid, and that's a good argument. However, what people who use that argument, when they fail to realize, is that the amount of stomach acid is very small. So if you drink a thimbleful of water with high pH, yeah, it'll get neutralized by the stomach acid. But if you drink like a glassful, the volume greatly overwhelms the volume of stomach acid. So the effect is pretty minor. Just a side comment.
Yeah, no, that's always been a big question of mine. Is there's the companies as well as I do that make alkaline water and sell bottled alkaline water, and the claims that they make.
I don't know them so well, but yeah, okay. But
yes,
in Japan, this is very popular, and I'm told that if you go to Japan, if you live in Japan and you have any gastrointestinal disorder, from your mouth to your rear end, anywhere in between, they'll pay for you to drink alkaline water, either to get a machine that produces alkaline water or bottle alkaline water. I'm not sure of details, but I've heard from several people that that's what happens. So there may be something to it, because I know they've done extensive tests in Japan, and I haven't followed the counter arguments in detail, so I'm not sure. Anyway, I make it a habit of trying to say nothing about any kind of commercial water, because I feel it's necessary to remain independent of any kind of product, any commercial product. We're doing pure science, and I don't want even to cast the impression that we're favoring something because they're paying us or something like this.
I can totally understand and appreciate that, sir.
Yeah.
And thanks for answering that question, because you probably shouldn't have. But anyway, so one more. I've done a lot of work with separating soil biology from different things, whether it be compost or soil. And part of the
You mean separating soil or soil biology?
Soil biology, so extracting biology from different matrices, everything from compost to actual soil. And then
how do you extract I don't understand. How do you extract biology from soil?
I'll tell you. So basically, I make a soft collider. So by that, I mean, if we were standing in water and I punched you, you would feel me. You'd feel the impact, but it wouldn't hurt. That's not true above ground or above water. So the concept was to brush organic matter together to peel off the humic fulvic acids, as well as any of the protozoa, nematodes, fungi that were attached to that organic matter. And then once you've extracted it, now you've got a solid and you've got this liquid that I lovingly refer to as chocolate water. And the chocolate water takes a long time for that fine particulate to settle out. So then now you have to de-water it. You have to separate all of the water from the solids. The solids can then go back to the compost pile or back into the ground. But by de-watering it, you're extracting pretty much all of the biology that you potentially could extract from that material. And in the dewatering process, I use vibration. I've tried presses. I've tried a bunch of different things. So this is leading to my question. Have you ever played with vibration and easy water in a tank to see if that did anything?
Well, yes and no. We haven't done exactly what you're suggesting, but we did try sound. We put a sound transducer in the chamber in which we grew easy water to see what impact it had. And we found at least frequencies around 100, 200, or 50 hertz, audio frequencies, low frequency. We found no effect. It was done in a fairly cursory way, and we never published anything. It needs to be repeated to see if there are effects, because other people are finding definite effects on plants and animals of using vibration, the frequency. I remember, for example, I remember in China, I was visiting China one time, and there was a symposium going on, and I just happened to show up at one of the presentations, and it was a Chinese group, and they were working in the forest. They identified two forest regions which were very similar to one another, and in one they played music, the other they played nothing. And when they played music, the plants grew in a more robust way. And there are people now who I would have a hard time finding the papers, but who are showing that some frequencies applied to plants can enhance their growth. And for me, if the sounds are enhancing growth, a good chance is that the sounds are having some impact on easy water. So I think it's a subject that needs further study. And again, we'd love to do it, but we can do only so much. And
we got to make you some more money so you can hire more students so that they can do the research.
Ken,
you're right on. You took that right out of my mouth, Ken. We got to find you more funding, sir. Yeah, and so the interesting thing was that in my experience with doing this stripping and dewatering, when I do apply the liquid back to the soil, I get aggregation almost immediately. So I'm clearly charging the organic material that does pass through the micron screen that I'm using at this whatever particular job I'm doing. Definitely is exciting the electrons and the protons, because otherwise they wouldn't just naturally pull the aggregates together so quickly in soil. Like, for instance, the sand and the clay platelets just seem to just right together real quick. So I gotta believe that it is definitely affecting the easy water that is in the organisms, in the biology itself, because we get explosive growth.
Well, it could be that, but when things aggregate, there could be a number of different reasons why. And in my fourth phase book, I discuss that in detail. For example, if you put two negatively charged blobs in water, you expect they're going to repel each other, but in fact, a number of people have shown, including us, that they come together, they attract one another, and it's completely counterintuitive. But the reason, and I'm not the one who came up with the reason, it was actually the great physicist Richard Feynman, who said, he called it like likes like. So if two like charges like each other, they come together. And that's what happens when you put the like charges in the water. They actually come together. You'd expect that they do the opposite, but they come together. And he said the reason they do that is so like likes like, so they like each other, because of an intermediate of unlikes. So you got two negative charge blobs, and if you got positive charges in between, they will attract, and that's what happens. And the reason you get positive charges, we found, oh yeah, there's a nice book. The reason you get the positive charges, as easy water builds around each of those negative particles, protons are released, and those protons are at high concentration between the two of them, and so they're pulled together. We've confirmed it. Other people have confirmed that it's totally counterintuitive, but the mechanism seems to be that.
So they get close, but they don't connect. They
can, depending on the situation, they can connect, but usually they'll be stable at some particular distance. But if you can imagine big spheres, they'll touch at one point, but on top of and beneath this touching point, you could have a lot of positive charges that keep the two spheres in contact with another. So it depends on it's a quantitative consideration. Usually they'll be at some distance apart, but sometimes they can touch.
Fascinating, my friend. Fascinating.
Yeah, well, it's fun. And all of this is described in great detail. And I don't know where you got that slide, but the book has turned out to be very, very popular. And I won't.
And there's a lot of other books on Amazon, guys. So it's and audiobooks, movies, etc. Like there's Cells, Gels, The Engines of Life. Gerald is well written, and you guys should all be reading those books.
And hopefully, when you buy them, you're getting some royalties, my friend.
Well, I'll tell you about that. So the publisher is me. We publish our own book. It's called Ebner and Sons, and Ebner is the nickname of my son. And the reason for doing it that way is if it's a scientific book, and if you look at scientific books, say on Amazon, you'll see them priced at $100 or $200. It means that the publishers make money on the number that they sell. And they don't sell too many because not too many people have it. And we're interested not in profit, but in dissemination. And so you'll find on Amazon the price is, I think, less than $30. And the reason, and it turns out that it is selling very nicely, but we don't really make money on this. It goes by my son is not only an artist, he's a professional artist, and he's great. If you flip through the fourth phase, you'll see wonderful art. But he runs he runs the publishing company too and takes care of all the stuff that I couldn't possibly take care of. And the other advantage of that is that we can publish it as we like. We can choose the format that's most appealing. We don't have to listen to what a reviewer says about, oh, you should remove this chapter because it's too speculative or something like that. We publish it as we like to publish it, and so if you look at the reviews, sometimes if I feel a little depressed on some day, I look at the Amazon reviews and I feel better.
That's awesome. Silly Lily, you've been staying one question ahead of me, which is fantastic. Yes, you're spot on.
Gary, I just want to check in. Are you good for time, or you want to have one last question?
Well, one last question is okay, and then I really got to go.
Thanks so much.
Why don't you take that? I've logged the mic.
If there's no questions in the audience, of course I've got hundreds, but is there one, Ken, or can I ask?
No, I have a million questions, but I'm sure we're going to bring Gerald back on as soon as we can convince him to do so, because this is an amazing conversation, but I'll let you ask the last question.
It may be a boring question, but I was just interested. You kind of mentioned something about UV and that easy water absorbs UV. What's the excitement there?
Well, okay. We not new anymore. A postdoc in my lab has been studying the effect of ultraviolet light on EZ, and we had some indication from older work that there might be something going on there, and she demonstrated it. What it does, UV light, and we studied around a wavelength of roughly 270 nanometers. What it does is increases the negative charge of the EZ. Doesn't expand the EZ, but it increases the negative charge. So basically, it imparts energy. But not by expanding the EZ, but by basically charging it up. In a nutshell, that's so we're trying to perfect the manuscript so that it's actually readable, and we're working on that right now. And so, thanks for that question.
Well, thank you, Gerald.
Okay, well, my pleasure. Thank you for the invitation. It's my favorite topic to talk about. Easy water, and I apologize that what I have said maybe is not so directly applicable to soil, but at least indirectly it has a lot to say in that.
I think there's a more direct correlation than you would like to believe, my friend, because it makes a lot of sense, and Av and I were talking about this when we looked at your research. A lot of things popped into our head going, well, this makes a lot more sense now on how plant reactions are, etc. So there is a direct correlation, my friend.
Well, I hear you, and thank you very much for that. What I really meant to say is that we haven't been focusing on soils and plants. I don't mean to say that I think there's no applicability of it. It's just that based on my training and my inclination and where I sit in the university, we've been focusing on animal cells mostly, not plant cells.
And again, bacteria. It's all about the biology, Ken. So
that too. Yeah.
So there's
all of biology, right?
It's a very deep connection.
That's my thing, Dr. Pollack, is it's all about the biology. And I learned that from Leighton-Morrison in the research he's done on the biology, and it all connects together. And we're going to have to find you some money so that you can start working on more experiments, because this is information that we really need to understand more about.
I hope you're successful. We really need the money. And I don't like to spend so much of my time, which I'm doing right now, trying to raise funds. It's enervating.
Aggravating, you said? Aggravating?
Well, that too. Aggravating and enervating. We really have so much, I think, going on. But, you know, when something is new, controversial, etc., it's difficult.
I get that.
Yeah.
15 years in the trenches trying to explain people there is actually life under your feet. MPK does not help it.
You understand very well.
I do, sir. I do. And I so appreciate you sticking it out for a whole hour and 45
Yes.
and letting us,
Is that what it is? Okay.
yeah, we've been
Thank you very much.
Okay, my pleasure. Okay, I got to get down to the mundane things. Okay, we'll see you guys. Thanks for the opportunity.
All right. Thank you so much.
Okay, take care.
Bye-bye.
Bye-bye.
So Silly, you're funny. You're one question ahead of me every time. The studies that I've seen, yes, too, show classical music for whatever reason has the right vibration to vigor plant growth. So spot on.
Is that Eastern or Western classical?
No, that's a good question. I don't remember what they said, but I would just tend to think that vibrate, you know, the strings and the horns are the ones that are really creating that that stimulation of what easy water, right? What else could be stimulating?
And to draw it back to you know, Emoto's work as well, and I think he showed with some of his pictures that I think it was Western classical music, you know, Brahms and Mozart and whatnot, that it did have a more crystal structure, more like the snowflake structure, and then heavy metal had more of a disturbance.
That's a nice way to put it, Ab, a disturbance. I've seen those pictures too.
And it parallels to what easy water looks like. It's got that hexagonal shape. So it could contribute a lot too.
Well, and you look at the work done with magnetism and magnets and moving different materials around in the forms that they've come up with. It's the same basic research, just on a different substance, and it all, you know, the more positive vibration that you're putting out, the better the structure and more beautiful that structure is.
And is it positive or is it negative? Now we're screwed.
Well, the earth is at a negative charge, and when we're grounding, it helps our bodies. So I think they're more the negative on that side of it, but only in balance. And it's really all about the balance.
And you know, that's funny. Those I can never pronounce his name. The Japanese water guy. I wanted to bring him up. Yamoto, thank you. And also whether he'd ever tried putting like a magnetic motor small enough into the easy water to see if that like prevented that magnetic connection that spun the motor.
Because have you ever seen that? I didn't run across that one in his research.
No.
I haven't seen it in his work either. But again, he's got so many friggin' experiments that he wants to do, and he just doesn't have the fucking money, which is, again, shame on the world, but we won't go there today.
Well, I think that's where the cannabis community, this is stuff that we really need to understand. And, you know, there's a lot of people out there that do have those types of funds. If they wouldn't mind, you know, donating a very small amount, it doesn't take a lot of people to add up to, you know, 1 or 2 or 3 researchers that he can hire and put in the lab to run these experiments.
Well, now we're a fundraiser.
For the biology, my friend, it's all about the biology, and we got to fix that problem. And I think this is part of the solution, and understanding it is what we need to be able to take to the governments, etc., to force them to go like, look, we have to change, or else, you know, we're not going to just not be here. We're killing us very quickly if we don't change.
Oh, well, that's pretty much obvious at this point. Again, I didn't want to go there in the conversation because it's just not pretty. But that being said, if if in your spare time, Ken, set up a GoFundMe and we'll start raising money on this channel and say, hey guys, come on, man, we got to get Jerry some more money. We got to get behind this, because in many ways it is everything, right? Water is life. So
Yes.
the work he's doing affects everything across the board.
Mhm.
So I think if there's any cause that could be considered the most crucial for humanity, the study of water would be it. Because, like, just thinking about cleaning, like one of the things in his book, he talks about scrubbing water and using these charges to push the pollutants out so that we would have clean, easy water to drink. So again, it's the foundation of all life. But now I'm getting heated.
Well, and consider when Kevin Jodrey was juicing the cannabis leaves, he was drawing that out as well. And is that what Korean natural farming is doing, drawing out, using the sugars to draw out the easy water from that cell structure? Is it all interlinked? Well, yeah, it is. At least as far as empirical evidence that I've seen from hundreds and hundreds of different people that are putting out content on this type of thing.
Agreed.
And Luna's work with juicing as well, right? So, yeah, I think it's
And Av, we got to get money for you too, so you can hire some more people in your labs and get more research done.
I think it's interesting because I think Elaine is one of the pioneers of how you can get marginalized when you are contradicting
Yes.
the norm. I mean, the idea that ATP may not be the driving energy force. Well, that's really going to affect the phosphorus fertilizer industry, wouldn't it? Right?
Just a little.
So yeah, it's going to be a bit of a challenge. And water is just viewed as a conduit, not an energy maker. And so it's definitely it's great to have people like Jerry and Elaine who are challenging the system,
and you and Leighton and a lot of others out there too.
Yeah, it's definitely hard to get the funding. So that's why it's a lot about getting out there and practicing it and truly, understanding it. So, getting out, juicing getting out there, taking, juicing our cannabis leaves for our own health, and getting out, as Leighton said, just getting out in the sun. And going for the saunas, like that infrared saunas or just any sauna, getting that and seeing how that feels. Grounding.
A lot of hot yoga places are using UV as well.
Mm-hmm.
UV or infrared?
Oh, maybe it is infrared. Maybe it is.
I think you can use far UV because far UV does not. And 270 is kind of in that far UV range.
Well, because when I've done hot yoga, they all have those red lights across the top. I don't know exactly what they're generating, but they're red.
Yeah, and that could be the infrared, and yeah, that'd be amazing.
Well, Av, do you have your copper plate under your feet right now as well?
I actually don't have my grounding mat.
What?
I was looking for it for my wife, and I mean, right now we're sleeping on a grounding mat, but
yeah,
we don't have the little one under my foot.
Bad boy.
Yeah, don't know. We got lost in the last cleaning. So.
It's where you haven't looked yet.
Yeah, exactly. Yeah.
All right, guys.
That's an oxymoron, Leighton. Now it's an oxymoron.
So next week, I'll see you in Edmonton. I'll probably be popping by the booth many times. But
we actually got confirmation we're supposed to have the OG's Power Hour and the Soil Matters in the consumption area. They're supposed to be setting us a table to do live interviews from there in the Bovita consumption area. So, you guys are going to be having to come there, and relax and consume and have a chat with us in those areas too.
So sorry, you will take live video and then upload it?
No, we're going to be live streaming the event. We can do it through cell phones. Oh. And so we'll be live streaming from the event for the 3 days and doing interviews. Not so much, watching the speakers give their talks, but doing interviews with the speakers, with the people that are have products at the event. We're going to be doing 3 days of interviews, basically.
We should ask our community who come and join us here to look over the speakers list.
120. We had Randy Rowe on the OG's Power Hour this morning. There's 120 speakers, guys.
Super. If they have a speaker there that they want to ask a question to, well, fire it to you, and
please,
and we'll track down the speaker.
And I'll have Av sitting there asking them, because he'll grab them and just haul them over. Okay, here, sit down, answer this question, please. Leighton, that goes for you too. What time are you arriving on Monday, Leighton? Are you going to be there early enough to do the show on Monday?
No, I don't get there till, I don't know, 4. And Av, how do I get from the airport to where you are? Is there a shuttle?
We'll probably either Uber or we'll find a volunteer to pick you up.
How about, okay, I got a volunteer. Her name is my wife. And, I think that that she would be more than willing to pick you up from the airport, Leighton, and bring you.
All right, so Av's got my itinerary. So just shoot it off to Ken, and then he'll know when I come in.
Perfect. I'll confirm that with Randy. But yeah, looking forward to it. Thanks so much for coming to Edmonton.
Coming out of my shell, you mean?
You might want to bring a mask. It probably is a little smoky up there. So
no, that's fine. Smoke doesn't affect me. Well, it does affect me in a positive way, unless I consume too much.
Yeah.
No, he's talking the wildfires that we have in Alberta. I can't see a mile down the road. There's so much smoke in the air right now.
Yeah.
Well, fortunately for you and I, Ken, we smoke cigarettes, so our lungs are used to being tortured, so it doesn't affect us like it does other people.
Yes, that's true.
No, seriously. I was talking with a guy who does high mountain training. He trains these elite military guys in high altitude training exercises. And he said, because you smoke, it would be easier for you to live in that environment or to adjust to that environment quicker. It's weird. And another thing too is people don't understand is tobacco is an antibacterial, antifungal, antiviral. So that's why I didn't catch the COVID, was because I smoke. And that's the only thing I can attribute it to, because I was around a lot of people that had it either before, after, or when they were supposedly coming down with it. So I don't know any other way to explain it, but there was a woman who was hired by the CDC to study the effect that the virus was potentially going to have on smokers. Mm-hmm. And she came back and said, don't worry about smokers. They're not going to get it. And even though the word was opposite and all the advertising, you're saying, oh, immune compromised people that smoke, you need to get the vaccines. You need to do this. You need to do that. And they stuffed her. They stymied her so that information didn't get out, because all people really had to do was get a nicotine throat spray, and they would have prevented them from it will get into your nose, but it can't get down into your lungs, and therefore it can't affect you.
Interesting.
But, yeah. And I have the paper somewhere that she cited about all of this. But no, they wanted us to take the vaccine. I'm sure everybody knows. Are we still live? Oh shit!
I better sign off before Neil Young gets upset with us.
Yes.
I will see you next week.
All right, peace out, guys.